Greetings everyone and welcome to my channel. In this video, we will be discussing one of the most popular tools used by bioinformaticians in differential expression analysis, DESeq2. DESeq2 is a bioinformatics tool that enables the identification of differentially expressed genes in RNA-seq experiments. However, not all genes are expressed equally, and some genes are differentially expressed in different experimental conditions. DESeq2 allows researchers to identify these genes and determine if they are up or down regulated. It is based on a statistical model that takes into account the variability in sequencing coverage between samples. The DESeq2 package is available on R software, which is a popular programming language for statistical computing and graphics. Firstly, we upload the following libraries, which are DESeq2, ggplot2, Heatmap, and enhanced Volcano. By using the package version function, we can get the version of the DESeq2. In our case, it's 1.36.0. And then we upload the count data file with read.csv function. As here we can see, in the data frame, we have 7 columns, starting by the gene ID, and then 3 replicates for the control group, and 3 replicates for the treatment group. And then we get the number of gene IDs inside the data with nrow function. In our case, we have 27,179 gene IDs. After that, we check if we have a duplicated gene IDs inside the matrix. We use sum to add or to make addition of the number of duplicated genes inside the count file. But we got 0 also. After that, we try to get the nice positions inside the data by using which is .na, and then we put the name of the count data file. We have 0 NAS inside the data. To perform DESeq2 analysis, a mapping of the columns of the count data to a corresponding group or condition is needed. DESeq2 uses a data frame where the node names of the data frame need to be the column names of the count data. The columns of the new data frame provide the mapping of the samples to a specific group. The first step is to create the data frame of the gene IDs. So in our case, in order to extract the gene names or the gene IDs, we write the name of our data or the name of the data frame and $, and then the gene IDs or the name of the column that we want to extract. And here we have the name of the gene IDs. The second step is to create the count data. Then the first column that we want to extract, which is the second column in the data frame, till the 7th column or the last column. As here, we can see that we want to extract the second column, which is the first of the control replicate, until the last column, which is the 7th column in our data. The third step to convert the count data into matrix by using as.matrix function. As here we can see, we check the class of the count data file before the conversion into matrix. By class function, it was a data frame. But after that, by using the as.matrix function, we convert it to matrix array. The provided R code is used to create a data frame called call data contains the information about the experimental conditions associated with the samples used in differential expression analysis. Let's break down each part of that code. Data.frame. This function is used to create a new data frame. Condition equal to as.factor C rep control 3 rep treatment 3. This part creates a column named condition in the data frame. The as.factor function is used to convert the function values into a factor variable, which is a categorical variable type in R. In this case, the condition values are generated using the rep function, which repeats the strings control and treatment 3 times each, creating a vector of 6 elements. Row.names, call names, count data file. This part sets the row names of the call data frame to be the same as the column names of the count data file object. The call names function retrieves the column names from the count data file object. Overall, this code creates a data frame called data with a single column named condition, which repeats the experimental conditions associated with the samples. The condition column contains factor levels, control and treatment repeated 3 times each, corresponding to the samples used in the differential expression analysis. The row names of the call data frame are set to the column names of the count data file object. This call data frame is typically used as a sample metadata when performing differential expression analysis to associate the samples with their respective conditions. After that, we go for prefiltration. While prefiltration low count genes prior to running DESeq2 in functions is not required, it is additionally beneficial for 2 reasons. By removing rows with very few reads, we reduce the memory size of the DDS data object and speed up the transformation and testing functions in DESeq2. Additionally, since features without any data for differential expression are not plotted, it can improve visualizations. In our analysis, genes with a total count more or equal to 10 are kept for the differential expression analysis. As here we can see, count data, row sums, and then the count data more than or equal to 10. The row sums command is a built-in R function used to calculate the sum of rows of a matrix or an array. This line creates a DESeq dataset object named DDS from the row count data, and the sample information called data. The count data argument represents the matrix of row reads counts, where the rows correspond to genes and columns correspond to samples. The call data argument represents the sample metadata, including the experimental conditions. The design formula condition specifies the experimental design, with condition being the variable representing the experimental conditions. This line simply prints the DDS object, displaying its content and summary information. As we can see, it performs the differential expression analysis using DESeq2 method on the DDS object. The DESeq2 function estimates size factors and dispersions based on the provided count data and applies a negative binomial generalized linear model to identify differentially expressed genes. The updated EDDS object contains the results of the analysis. In order to save the normalized tree counts, we use the counts function, and then we put the DDS data and normalized equal true. And then with the right.csv function and the name of the normalized counts, and then the normalized counts.csv file. Then we use results function for the DDS data. This line extracts the results of the differential expression analysis from the DDS object. The results function is used to calculate the log-to-fold changes, p-values, and adjusted p-values using the Benjamini-Hochberg method for each gene. The contrast argument specifies the comparison of interest, in this case comparing the treatment condition to the control condition based on the condition variable. The alpha argument sets the significance threshold to 0.05, controlling for false discovery rate. The resulting object trace contains the differentially expressed genes along with their corresponding statistical measures, which can further explored and analyzed. Then we use the summary function. The summary function is to get the number of up and down regulated genes. 21% of the gene IDs were upregulated, whereas 24% of the gene IDs were downregulated. PlotMA visualizes and identifies gene expression changes from two different conditions. In our case, it's treatment versus control. In terms of log-fold change, M on y-axis, PlotMA visualizes and identifies gene expression changes from two different conditions, treatment versus control, in terms of fold change on y-axis and log of the mean of normalized expression counts on x-axis. Here, by using plotMA function, we can plot the MA graph. Generally, genes with lower mean expression values will have highly variable log-fold change. The blue dots are representing the differentially expressed genes, and they have adjusted p-values of less than 0.05. The triangles in the edges of the plot are genes with higher fold changes, as the direction of the triangle are the direction of the fold change. For example, this gene was upregulated, whereas these genes were downregulated. Genes on the right part of the plot here means that these genes have high mean of normalized counts and high fold changes, which are very interesting to look into. Shrinkage of effect size is useful for visualization and ranking of genes. It's more useful to visualize the MA plot for the shrunken log-to-fold changes, which remove the noise associated with log-to-fold changes from low-count genes without requiring arbitrary filtering thresholds. Here, by using LFC shrink, and then we put DDS, condition treatment versus control. And then plot MA, res LFC plot MA function. After that, we perform the dispersion plot. Dispersion is a measure of spread or variability inside the data. Variance, standard deviation, interquartile range, among other measures, can all be used to measure dispersion. However, DESeq2 uses a specific measure of dispersion related to the mean and variance of the data. When, for example, we have 0.01 dispersion in our data, it means that there is 10% variation around the mean expected across the biological replicates. DESeq2 dispersion estimates are inversely related to the mean and directly related to the variance. Based on this relationship, the dispersion is higher for small mean counts and lower for large mean counts. The dispersion estimates for genes with the same mean will differ only based on their variance. Therefore, the dispersion estimates reflect the variance in gene expression for a given mean value. We have 3 steps for estimating dispersions. The first step is get dispersion estimates for each gene by maximum likelihood estimation. Second step is to fit a curve to a gene-wise dispersion estimates. This curve represents the expected values of a dispersion, gives the expression values of the genes. Last step, shrink gene-wise dispersion. Estimates are shrunken towards the curve to get the final dispersion estimates. Some genes will not be shrunken towards the curve because the variability or dispersion is so high. So DESeq2 will assume that these genes will not follow the modeling assumptions to avoid the false positive that may happen because not fit for the model. And by using plot this P estimate, we can get the dispersion plot, which is dispersion on the y-axis and mean of normalized counts on the x-axis. Then, principal component analysis plot, PCA. The PCA describes the variance in the data as it shows if the samples are connected to each other or not. PCA1 is the most variation in the data, the gene expression across the 6 samples, and PCA2 is the second most variation in the data. Before performing the PCA plot, we have to make the log2 transformation of the normalized recounts in order to improve the clustering for the visualization. As from the chart, we can see that 3 samples are on the left side of the graph and the other 3 samples are on the right part of the graph. To understand what their clusters mean, let's know how big is the variation in the data PCA1 represents 4. When using these unsupervised clustering methods, log2 transformation of the normalized counts improves the distances clustering for visualization. DESeq2 uses a regularized log transform of the normalized counts for sample-level quality control, as it moderates the variance across the mean, improving the clustering. Blind equal to false should be used for transforming data for downstream analysis, where the full use of the design information should be made. Blind false will escape pre-estimation of the dispersion trend if this has already been calculated. If many of the genes have large differences in counts due to the experimental design, it is important to set blind false for downstream analysis. When blind false, the experimental design is only used by the R-log transformations in calculating the gene-wise dispersion estimates in order to fit a trend line through the dispersions over the mean. Only the trend line is used by the transformations, not the gene-wise estimates. This code creates a volcano plot for visualizing the results of the differential expression analysis, which typically contains information such as log2-fold changes, p-values, and adjusted p-values for each gene. Lab equal row names for S for results. This specifies the row names of the results object should be used as labels for the data points in the volcano plot. X equal log2-fold change. This indicates the log2-fold change values from the results object should be plotted on the x-axis, whereas y equal p-value indicates that the p-values from the results object should be plotted on the y-axis of the plot. Title, treatment versus control. This is the title of the plot. P cutoff. This specifies the significance cutoff for the p-values. FC cutoff equal to 0.5. This sets the fold change cutoff for log2-fold changes, and point size equals 3. This sets the size of the data points in the plot. Lab size equal to 6. This sets the size of the labels for the data points in the plot. Now let's perform the plot. As here we can see, these genes were upregulated, while these genes were downregulated. After that, we go for data quality assessment by sample clustering and visualization on heat maps by the following codes. This code performs hierarchical clustering and generates a heat map to visualize the distances between samples based on their gene expression profiles. Let's break down the code. Sample dests equal to dest TSA RLD. This line calculates the pairwise distances between samples based on their gene expression profiles. The assay function extracts the gene expression values from the RLD object, which is likely a transformed version of the raw gene expression data. The T function transposes the gene expression matrix so that samples are represented in columns. The dest function then calculates the distances between the samples. Library rColor-Brewer. This line loads the rColor-Brewer package, which provides color palettes for visualizations. Sample dist matrix equal to s.matrix sample dist. This line converts the sample dist object, which represents the pairwise distances between samples, into a matrix format using the s.matrix function. The resulting matrix is stored in sample dist matrix variable. Regarding column names, sample dist matrix, this line retrieves the column names of the sample dist matrix, which correspond to the sample labels. It simply prints the names of the columns. Colors equal to color ramp palette ref_brewer.pal 9 blues 255. This line generates a color palette for the heat map. It uses the blues color palette from the rColor-Brewer package, which consists of 9 shades of blue. In this case, it creates a gradient of 255 colors. Heatmap, simple dist matrix, clustering distance rows equals sample dist, clustering distance calls sample dist, color call equal colors. This line generates the heatmap using the pheatmap function from the pheatmap package. The sample dist matrix is passed as the input data for the heatmap. The clustering distance rows and clustering distance calls arguments specify the distances used for hierarchical clustering of the rows and columns, respectively. In this case, sample distant object representing the pairwise distances between samples is used for both row and column clustering. The call argument specifies the colors to be used in the heatmap, which were previously defined in the colors variable. Overall, this code performs hierarchical clustering based on sample distances and generates a heatmap visualizing the distances between samples. The resulting heatmap provides insights into similarity and dissimilarity of gene expression patterns among samples. As we can see from the graph, the color represents the distance between the samples. Treatment samples look different from the control samples. The dark blue diagonal line here represents the high similarity between samples because it's the intersection of the samples with themselves. For example, treatment 2 with treatment 2, or treatment 1 here with treatment 1. In addition, treatment 2 is more or less close to treatments 1 and 3, as all appear dark blue color. Example here, treatment 1 is treatment 2, whereas control samples are light blue. Treatment 1 with control here, light blue, which represents the high dissimilarities between these two samples. Then a heat map of the log transform normalized counts for the top 20 genes was generated. Let's break down that code. Top genes equal to results, order, results, $, and p-adjusted value for the first 20 genes. This line selects the top 20 differentially expressed genes from the results object, which likely contains the results of a differential expression analysis. The order function is used to sort the rows of results based on the adjusted p-values in ascending order. Results object is then subsetted to select the top 20 rows or genes, and then top genes equal to row names. This line reassigns the top genes variable to contain only the row names of the top genes object. Heatmap assay RLD top genes, cluster rows equal true, show row names equal true, cluster columns true also, font size row equal 8, and annotation code equal the call data. Let's run this code. As here we can see, we have a heat map for the control samples and the treatment samples. We can see that there is a big difference between the control samples and the treatment samples based on this color scheme. Overall, this code selects the top differentially expressed genes based on the adjusted p-values, extracts their expression values from a normalized dataset, and generates a heat map to visualize their expression patterns. Then, a heat map of these scores for the top 20 genes was generated. A z-score of a 0 indicates that the gene's expression level is the same as the mean expression level across all samples, while a positive z-score indicates that the gene is expressed at a higher level than the mean, and the negative z-score indicates that the gene is expressed at a lower level than the mean. The provided R code calculates a z-score for a dataset using custom function and then subsets the z-scores based on a selected set of genes. Let's break down this code. The first one is cal z-score function x to x minus mean x over standard deviation of the x point. This code defines a custom function named cal_z_score. This function takes a vector x as input and calculates the z-score for each element in the vector. The z-score is calculated by subtracting the mean of x from each element and dividing it by the standard deviation of x. Z-score all, t apply normalized counts, 1, cal z-score. This line applies the cal z-score function to each row, which is the genes in the normalized counts matrix. The apply function is used to apply a function, cal z-score, along a specific dimension, 1 for rows of the matrix. The resulting z-scores are stored in the z-score all matrix. The t function transposes the matrix, swapping rows with columns to have genes in columns and samples in rows. Z-score subset, Z-score all, and then top genes. This line subsets the Z-score all matrix by selecting only the rows corresponding to the genes specified in the top genes vector. The resulting subset is stored in the z-score subset matrix. In summary, this code defines a function to calculate the scores and applies this function to each row or gene of a normalized gene expression matrix. The resulting z-scores are stored in matrix z-score O. Then a subset of the z-scores is created by selecting only the rows corresponding to the genes specified in the top genes vector, resulting in the z-score subset matrix. The z-scores provide a standardized measure of the deviation of gene expression values from the mean, allowing for comparison across genes and the identification of potentially significant changes. Thanks for watching. I hope you enjoyed this video. If yes, then please don't forget to subscribe to our channel, hit the like button, share the video, and leave your comments under the comment section. Until next time, happy analyzing.